In the present day, clinical research and data science are heavily relying on large sample sizes consisting of hundreds, thousands, or even tens of thousands of patients. And even for each patient, we're starting to deal with large, diverse data sets of potentially complex temporal relationships and various associations. We're also becoming to rely on systematic data processing, including custom algorithms for data analysis, such as radiomic feature extraction and the application of machine learning technologies. Despite this need, we lack a common platform for conducting this kind of research. Our data sets extend across various clinical sources and repositories, often requiring significant manual work to retrieve and collate. Individual labs typically develop their own analysis pipelines. Often this is built from disparate chunks of MATLAB code, working from Excel spreadsheets of data. This work is often therefore performed in isolated silos, leading to significant duplication and waste across various teams and departments within the organization. Further, these prototypes are typically unsuitable for prospective trials since they're sitting on a student's workstation in some hobbled code. They lack robustness and proper testing, and they offer little to no integration with our clinical systems. Moreover, they often have poor to non-existent user interfaces. This all leads to limited capacity to perform large-scale systematic research at a leading institution such as ours, using the wealth of data that in theory we have access to. In response to this need, a group of us in the radiation medicine program set out a couple years ago to develop an agile and modular software framework that integrates with key clinical systems to facilitate large-scale automation, data science, clinical research, and essentially prospective decision support. The name of the system that we developed, we call MIRA. I know it doesn't stand for anything, and recently we came up with a joke that MIRA stands for MIRA isn't really an acronym. But I'll take a few minutes now to describe some of the main components and features, which collectively we think provides immediate and future value in terms of enabling the type of research that we're talking about here. In a nutshell, MIRA is a software toolkit. It's a set of libraries that researchers like ourselves can use to build data mining, automation, and analytics applications. It is built up from a stack of software interfaces, which are essentially little components that provide integration with the various clinical data systems and data sources that we have access to. It also provides a customizable image processing pipeline and a dynamic, flexible database. So all the building blocks we need to deploy research pipelines. And I'll discuss some of these components, some of the specific ones that have led to some of our first applications. The first and most notable component of MIRA is a software interface with RayStation, which is our radiation treatment planning system in the RMP. RayStation is sort of the key piece of software that we use clinically to drive all of our patient treatments. And for RayStation, we built a versatile software module that can extract essentially any of the data within RayStation for a given patient. So this includes the radiation treatment plan, which consists of a description of all of the treatment fields, the radiation fields that we use to treat our patients, the 3D dose distributions, which define how we're actually depositing the radiation energy within the patients, ROI geometries or the other annotations that our clinicians and support staff make on this data, image registrations that align and associate different data sets, and accumulated dose, which is a description of the actual treatment that we deliver over the course of therapy, which might deviate from what we thought we were going to deliver. But it's not just about the data extraction. We also establish two-way communication or control with RayStation, and this allows us to leverage all of the RayStation services for the automated pipelines that we wish to build, such as auto segmentation, automated planning, dose calculation, deformable image registration, to list some of the technologies that we can now take advantage of. We've also built another powerful interface. This one is with our oncology information system, Mosaic. And this is a major source for all of our radiation delivery data. So all of the data that surrounds the actual delivery of the treatment, the scheduling, the timing, as well as some diagnoses and staging and other clinical data. So this also includes which plans and fields were delivered to a patient and when, on what day, at what time, as well as other treatment data such as daily cone beam CT or MRI images, so volumetric images that we acquire of the patient every single day that we treat them over the course of their treatment. With this information, we can automatically gather and assemble the full data trajectory for a given patient over the course of their radiotherapy, which could last 5, 10, or 30 treatment fractions long. Another important component of this system is our custom database. This was built using some non-relational technology known as MongoDB. This is open source technology, and it's designed to support flexible, scalable data models with arbitrary structure. Essentially, what this means is that we're not confined to the particular tables or columns within an Excel table, and that we can build out complex data relationships that belong to our patients, and then use this in this programming environment to build these pipelines. We use this database as a standard storage system, of course, to store, let's say, image features that we compute, but we also use it as an advanced buffer for transferring data between the various modules. So we might extract data from one system, store it in the database, and then queue it up again at a later time point when we're interacting with a different system. And so within this data store, we maintain all of the basic information that we're used to working with our patients. Lists of patients, their images, their dose information, outcomes or other toxicity-related information, as well as cohorts or lists and groupings of patients that we can manage. And we also design this database to be easily extendable with arbitrary types of data. So collections of, let's say, radiomic features or other derived data that would be specific to given studies or certain applications or research pipelines that we build and support. Another major feature of the platform is this analysis module and scientific computing environment. This is essentially a sandbox. It's built on the Python programming language, and it has some core scaffolding that leverages several open source computing packages. And this allows users or pipelines to analyze any of the data that we access by the system. So if we connect to a clinical system like a PACS or our oncology information system, we grab these images, we provide all the scaffolding to take that into the research environment where users can plug and play with their algorithms. And it's really in this environment where the meat of any image or data processing occurs. And this is where the extensibility of the platform really shines. If a research group has a new prototype algorithm or a prediction model, say a deep neural network they built using Google's TensorFlow, and now they wish to test it against a cohort of patients sitting in our diagnostic packs, or even applied in a prospective study, we can simply embed that block of code and rapidly deploy it without this scientist or an investigator needing to worry about any of the other implementation details. And lastly, there's this distributed web application. So essentially, we've built up the infrastructure to support web apps for displaying or visualizing any of the data within MIRA. So essentially, this is an interface that you access through a browser. It's a web app through a secure connection anywhere within the organization. So anywhere within UHN, you can access this web app. We can build custom visualizations or dashboards that are tailored to specific projects. And we can do this very quickly. And I'll give an example of one of those in a moment. But ultimately, our vision is that through MIRA, we can build custom reusable pipelines or black boxes, so to speak, that bury the complexity of the analysis and allow our users to quickly inject data and automatically retrieve their desired results or output. This could mean specifying a list of 1,000 cancer patients whose data are living somewhere within the UHN data sphere and instantly generating the parameters for a given prediction model, or perhaps compute a library of 1,000 engineered texture features on this cohort. Or take a predefined, pre-built pipeline and generate real-time toxicity assessments for use in a prospective clinical trial. So I'll move on to discussing a real-life example. Radiation pneumonitis is a dose-limiting toxicity in non-small cell lung cancer that affects up to a quarter of all patients. In clinical practice, the risk of a patient developing pneumonitis is estimated using standard dosimetric and clinical parameters. In a previous study, through collaboration between Dr. Drew Hope, a radiation oncologist here at the Princess Margaret and in the audience with us today, and in collaboration with a team of Danish physicists at Odense University, it was previously shown that an image marker that was derived from cone beam CT images, which are images we acquire daily of our patients, is correlated with radiographic pneumonitis in patients treated for this disease. The marker that was designed in this study essentially measures the amount of normal lung tissue that exhibits a significant change in density during treatment. So essentially, they're looking at healthy regions of lung and looking to see how opaque it gets over treatment as the radiation is delivered. And using deformable image registration to align and correlate tissue across various images, this marker is computed by assessing the density change at fractions 10 and 20 throughout a treatment course lasting 30 fractions. So we're talking about 1/3 and 2/3 of the way through treatment. An initial pipeline was prototyped in MATLAB by the Odense group and was implemented here locally. And one of our goals was to re-implement this process in a fully automated fashion using MIRA, using our clinical systems to demonstrate that we could take what was on a grad student's computer and implement it right within our clinical workflow. This would then enable us to analyze a larger number of patients and fractions, but to also be directly deployed as a tool in a future prospective study. So I'll just briefly take you through the actual pipeline and how this image marker is computed using the MIRA pipeline. So the process begins with an initial radiation treatment plan and three-dimensional dose distribution in our radiation treatment planning system, RayStation. Then treatment begins, and as the treatment course progresses, we configured our clinical treatment our clinical treatment delivery machines to export these daily cone beam CT images to our oncology information system and automatically transfer these images into RayStation as soon as they become available. So we have something that's watching the oncology information system. It can detect as soon as the radiation treatment is delivered, since all of our systems are connected over the network, and then immediately take these images and bring them into RayStation. Of course, for our retrospective studies, we skip this step and instead reroute our system through DICOM archives to access the images. Then we take automated control of RayStation, and we begin a series of image registrations. We register and align the cone beam CTs with the planning CT according to how the patient was aligned at the treatment unit. And for this, we also grab additional information from the oncology information system. This defines how the patient was set up at the treatment unit and how they were aligned relative to that first radiation treatment plan. Then we start performing automated deformable image registration, focusing on the lung region so that we can align individual voxels, individual pieces of tissue between subsequent cone beam CTs to assess that density change. So once we rely on RayStation to do that registration, we then pull all of this data out of RayStation and transfer it within our analysis environment. And in there, we create regions of interest based on both the dose distribution and the anatomy, and combine these to create sort of an analysis mask. And it is within this mask that we look at the difference in the cone beam CT image values, construct a histogram, and essentially compute the image marker. Finally, the results of the analysis are saved in a custom collection in that database, and then we can then visualize and review this in real time on the web application. And that's a screenshot of the actual web application showing a chart of this cone beam marker progressing over time. From the acquisition of the cone beam CT at the treatment unit to the serving of the data to the web application, the entire process is automated and takes only a couple minutes. So here are some results comparing the automated workflow to the previously established manual workflow that was using MATLAB. On the right, we have some correlation plots that essentially just show the agreement between the marker values computed using MIRA and the manual MATLAB approach. So these aren't just using two different softwares to perform the process, but there's actual differences in the registration algorithms that are used to derive the markers. So there's not 100% agreement, but the agreement is quite strong. But looking at the table here, we also see that the automatic pipeline is both faster and more reliable than the old method. So using MIRA, we were able to successfully process nearly all of the patients compared to only 2/3 previously. So the manual pipeline just broke down for 1/3 of the patients after fighting with the data for several hours. We also reduced the processing time from roughly 55 minutes per fraction down to only 2 minutes. And that's 55 minutes per fraction of time spent by a trained expert versus 2 minutes of unsupervised computation time. And so with the manual approach, that meant that we really only had the bandwidth to look at 162 images in total. And with MIRA, we easily expanded that to all images for all these patients, which meant 3,500 just in this cohort of 100 patients. Using these additional patients that we were then able to process with MIRA, we were able to expand on the previous work and carve out an independent validation cohort to demonstrate that this marker not only predicts for grade 1 pneumonitis, which is essentially just post-treatment radiographic change in the lungs, but we can also selectively predict for the more clinically interesting higher grade complications as well. So that's grade 2 and above symptomatic pneumonitis. Pardon me. Because the processing is fast and automated, we're now able to calculate the cone beam CT marker for all 30 fractions throughout the given treatment course. So before, just fractions 10 and 20 were selected because, again, we had limited bandwidth in terms of the manual approach. With the ability to measure the marker at all daily fractions, we're able to track the changes in this marker over time at a high temporal resolution. And we've demonstrated that patients who develop grade 2 or higher pneumonitis have a consistently higher cone beam CT marker throughout their treatment. And the heat map below, for example, shows an example of a rolling toxicity prediction for one of our patients who went on to develop symptomatic pneumonitis. So with this technology in place, we'd like to take another leap forward and now test these models in a prospective setting to predict the pneumonitis risk for each patient every day over their treatment course. And this is something that we're planning now. Ideally, however, we'd like to do this even through a multi-institutional clinical trial so that we could capture and test the model on as many patients as possible. Unfortunately, deploying this machinery at several institutions across the country is, of course, pretty challenging. This is still a prototype, and it relies on somewhat bespoke software components that we've designed here in-house. So how then can we hope to leverage this technology on a larger scale? And that's where QIPCM enters, which stands for Quantitative Imaging for Personalized Cancer Medicine. Many of you here in the OICR may be familiar with QIPCM. Essentially, this is a combined research group and infrastructure project funded by the OICR. And QIPCM fundamentally aims to provide end-to-end data management and support for both local and multi-institutional clinical trials. So QIPCM provides a centralized hub for aggregating and analyzing data for multiple sources across various institutions, as well as within UHN, providing a common platform for image analysis and review. So they have databases and servers that you can log into and use some common tools to both analyze your data as well as review your data. And we've now teamed up with the QIPCM project to embed the MIRA framework within their infrastructure. So this is a busy slide, but in the green box there is sort of the MIRA box that we're now working to integrate within the entire QIPCM environment. By doing so, we're hoping to operationalize the MIRA automation pipelines in a robust and reliable way that can then be accessed by any study that uses QIPCM. There's something like 35 ongoing active clinical trials that rely on the QIPCM infrastructure. This would allow us to quickly deploy custom image processing pipelines, such as the cone beam CT marker for pneumonitis prediction, in such a way that any institution could process their cone beam CTs, for example, and generate these toxicity predictions immediately for their patients. In this light, we're now designing a much more generic, extensible radiomic feature extraction pipeline within QIPCM that could be extended to any radiomics or image processing study, not just the cone beam study that I mentioned previously. This pipeline essentially allows researchers to select patients. So that's sort of like the first step based on predefined cohorts. So we can grab patients that belong to a particular study, but we can also access the clinical systems and automatically group patients based on some of their clinical data or other features of their treatment data, such as their diagnosis staging or other clinical factors. Then based on a programmable analysis schema, the system automatically extracts the relevant images from whatever the data source is. So it could be the JDMI PACS, for example, or the QIPCM data store, extract the relative or the relevant images, dose, contours, and pipe this through the MIRA analysis environment. And that's where all that number crunching goes on, where the custom features are calculated, they're time-stamped to the images, and they're inextricably attached to those images in the database. And then we can store this data and serve it over our web app for visualization, exploration, or interactive modeling, or we can export it back to the investigators for further modeling and analysis. And of course, there are many studies nowadays that are exploring predictive radiomic markers, but up until now, there's been limited work to directly integrate these into clinical care or multi-institutional trials. So we're trying to jump the gun here, and through our collaboration with QIPCM, build a robust automated pipeline that can go seamlessly from model development to deployment in clinical practice, all within a single environment. This platform will allow for individualized image marker tracking from diagnosis through to their entire treatment course, which is an important step towards personalizing treatment based on real-time imaging feedback. And that's the ultimate goal of this work. So thank you for your time. And I'd like to take a moment to thank our core team and our collaborators here at UHN and at QIPCM and in the radiation medicine program. Thank you.